So what is this COVID-19? We have heard enough, we have known enough, but seems to be really an enigma. We don't know enough to control it, but we seem to know a lot. So this is caused by a teeny virus, which is just 30 kilobase RNA. It's a very small RNA, just 30 kilobase in size. It's a positive strand RNA called SARS COV-2. It was originally called as Wuhan virus. Those are all changed now. So what is this sequence of events? Firstly, one gets exposed to the virus. Not necessarily the exposed people always get into the next step of contracting. Here is this contracting. This is contracting, that's the upper respiratory tract. The virus goes and sits and start dividing. This is contracting. A lot of people, actually nothing happens to them, because if any exposure will get us into contract, we would have all contracted long ago. There is repeated exposure, it's a prolonged exposure, the dose, the viral dose or the numbers. See, that's why wearing a mask, avoiding crowd, keeping safe distance, using sanitizer, all of them helps despite exposure, because your exposure becomes lesser and lesser dangerous if you can actually do it well, in the sense unnecessarily close contacts if you avoid. Then you get early symptoms. This is maybe in a week's time, 4 days to a week's time, you get early symptoms. Then it can either go into a more serious infection into lower respiratory tract, and then there is a mortality rate of 2 to 3%. See, this changes. Some cases it's even less than 1%. In some demographics, it can be much higher. Or after early symptoms, one can actually completely get cured as well. So throughout this phase, diagnostic methods are very important. See, you might have heard the WHO and other folks telling, no, test, because, and there are so many different tests. What you will do when, I think I'll try to explain before we get into the main point. So what is this? Here is our virus. You can see that the virus also packs not the full-length RNA. Apart from the full-length RNA, the virus does packs smaller RNAs. That's why sometimes no S gene or E gene is giving better signals, or a lower CT. I'll also tell what is this CT is all about. So with this, we can move ahead, because here is the virus. You can see it's made of different proteins. Those are used as target for the antigen tests, right? The spike protein is a major target for most of the antigen tests. And the RNA is here, and then the N gene is also being used for antibody tests. They might use S gene or N gene for the antibody tests. So here is all a very little target for us. It's 1 in million of us, or even smaller. So why is diagnostic important? Firstly, we have to identify. Simply because I'm having cough or fever doesn't mean that I'm having COVID, right? So it's important that we have to make sure that it's correctly identified. Otherwise, we'll panic. And the next important thing is contact tracing. I'm having COVID. Where did I get it from? Maybe from a taxi which I took yesterday, or maybe not. So how will I find him and stop him? That's important, because that person also has to be tested. Whether he should do an antigen test, RNA test, or antibody test will come. And then next is containment. Okay, I'm now known to have COVID. Let me be in my home, and I don't meet even anyone at home. Maybe I'll cook myself, or people will push food to me, and I isolate myself for a week's time. That's all is important. So finally, how do you prevent? How do you treat? All of them, diagnostics is important. So if we don't diagnose, who will we treat, right? And what will we do? So these diagnostic tests have become really, really important. Before I get inside, very quickly, what are the terminologies? See, diagnostics, coming from biochemistry, I can say diagnostics is an absolute science. I have to say, do you have COVID or you don't have COVID? That's it. Whether my blood glucose is 100 mg or 400 mg per ml. Whereas screening is different. Boss, you have slight pot belly, maybe you're obese, maybe you'll have this, maybe you have that, right? So screening, maybe I'm having cough, I'm feeling feverish, I'm feeling tired. So screening is less of a science, but it should be still amongst 100 people, I can find out those 10 people to whom I will do the test. So a screening test need not be accurate. It can have false positive, right? But a screening test should actually narrow down. That's more important. If everyone is positive, a screening test can't do that. And what is false positive test? That is, I do a test and then I'll see that that person is having COVID, but actually is not having it. The test has wrongly interpreted something is false positive. And you always hear this false negative. That is, if you do a test, if I'm exposed today, and tomorrow if I do a test, I will show negative. And day after tomorrow, I'll show positive. People wrongly say that yesterday's test was false negative. There is a time it takes. So on a same day where qRT is positive, antigen test might be negative. Antibody test will take at least 10 days to show up. So you can't say that antibody test was false negative on the day 3, right? So there is, but a false negative could be that I used a kit A which showed positive, but a kit B of qRT did not show. Maybe a lab, one lab is, so it's false negative. Everything is actually it's more of a sensitivity issue. Okay, it's a sensitivity issue. How sensitive is your assay, right? Vis-a-vis how much is the viral load or the RNA load. Then is specificity. Am I getting, even if I'm having flu, if I use a thermometer to check myself, then I will say, like, boss, you are having COVID. No, it's a temperature measurement is not specific to COVID. It can be used as a screening method, right? So an antibody test or a qRT test should be specific to COVID. It can't say influenza virus or some other virus or maybe even host gene. It cannot. So now the current tests are specificity wise good, sensitivity wise reasonably good. Different kits have different sensitivities, of course, you know. And the next one is called concordance, right? Because see, usually biochemistry is never done on concordance or any test. One should never use this word concordance at all. But because of this pandemic time, people are saying, look, I have 20 samples, 10 of them are negative, and the rest 10, I have different levels of the virus, right? That is measured by CT. I'll tell you, I think you all know what CT is, but I'll tell you what CT is all about. Okay. So if it concordance is good with my kit and whatever I know, I say, hey, this kit is good to go. That's an easy way of approving a kit. I think most of the kits today are done by concordance tests, assuming the manufacturer has done every test properly. So it's important if you're manufacturing, right? Concordance is not the right way, but that's the best possible alternative to a rigorous test. Finally, prognosis, because prognostic tests are different from diagnostic tests. Because, for example, the viral load, people will say, hey, if you have higher viral load, maybe it is severe, or maybe not. So prognostic tests are different from diagnostic tests, which I'm going to come in the next few slides. But prognosis is something which will tell, I'm having COVID. Can I just isolate myself and watch TV, watch the news, Or should I go and get myself admitted into a hospital? Or should I get into a ventilator? Right? There are many things. All of them you can call it prognostics. The best test is culturing the virus. Sorry, who's doing it? Right? ICMR was initially doing it. And then proving that that is the COVID virus you guys are having, right? It's too dangerous to do it, right? So we can't do that anyway. So I think I sent to two sets of people two interesting things. First is, what is that non-invasive test that can become invasive if you don't cooperate? I hope, guys, you all guessed it. This is the test. People are using dogs for testing. Actually, it is screening, right? So if you don't cooperate, it can bite you. That becomes invasive. This is the first one I said. Second one is, I said there is a case of a diagnostic test, which is also a medicine. I hope many of you would have guessed it, right? It's a simple test. You can all do it at home. I hope many people are doing. If you don't like it, you can use wine. So this will test you for smell and taste. And it's a good test to do. That's our good old monk. Old monks can try this. So as long as you can smell old monk, as long as you can taste, maybe it's not COVID. But look for other symptoms, please. So now let's go back into serious things. Some examples of diagnostics. This is a rapid antibody test. I hope I can show you this test. See, I'm holding one of the cards. This is a negative test. You can see the control band coming up, but not the test band. Here is a positive. I'm sorry, here's the positive test, where you can see the test band as well as the control band. Both of them are showing up. Here is my friend in Gujarat. He's manufacturing these tests. So you can see here, this is a IgM, IgG, and so you can see positive case. Or you can go to lab and do a ELISA test. Or here is a diagnostic PCR test. This is what we are all going to talk more about today, is this diagnostic PCR test. And then comes the antigen test, which is also like this card which I showed, antigen card. Then comes the LAMP test. This doesn't need a PCR machine. It starts with the RNA. It's colorimetric test, which you can see by eyes. See here, this is the negative control, no template. This is the positive control, which is one of the target COVID RNA added. And here is the internal control. This is a human gene. And then the test. This can be done in less than 30 minutes time, by the way. So, and you don't need any particular equipment here. You need only a 65-degree water bath, which you can set it up with a small, this thing, no? Even in your home, you can set up this in a water bath and a thermometer, you can do it. And then comes CRISPR-based diagnostics. I'm just bringing it because this is the most modern technology. As you know, Tata and IGIB CSCR Institute in Delhi, together they have developed it, and I understand it's going to be launched very soon. It's ICMR approved. So here the RNA is converted to DNA, and then CRISPR is used to find out. This is like a rapid, this is a paper strip, so it's cheaper. So this is the control. And if you see the next band, then you have got the, so that's the positive. So if someone is having COVID, so it's a qualitative test. So this is a negative patient or a negative person. So you will see the control band, you will not see the COVID specific band. And then there is Sherlock Bioscience. This is a US based company, sold only in US. There they take the RNA and convert into a RNA again, using make a cDNA T7 polymerase. And then they use the classical, they use that as a bait for a classical CRISPR to work on. NGS is also used. You can Illumina, you know, as you know, the machines are expensive and you need 3-4 people to operate the machines, but they can do maybe 1,000 samples a day easily. Nanopore sequencer, which we are, Venkatesh, as Venkatesh told, which we are having and we are also distributing, has come up with something called as LAMPORE, that is LAMP assay and using the pore. So one can run 1024 samples now, and it is scaling to 4096 samples in a day, right? This is something amazing. This can make a huge difference, I tell you. And it's a small machine which one can use, and it cost comes down, capacity goes up. It's as sensitive as qRT. This is a replacement maybe coming for qRT, I don't know. So just before we go into our main thing, host monitoring is also important, like temperature, cough, taste and smell. I told you, if someone is really having fatigue or doing well, and whether there is a symptom or no symptom test is there. Once COVID is confirmed, there are different tests, liver function tests, platelets, and blood cell types. That's very important. And many people, if they get COVID, the blood clotting time reduces. That's why you can see they have to be treated with blood thinners. Otherwise, they can get a block, maybe a heart attack or a stroke can be the result of it. CT scans, oximeters, that's to monitor the host after one gets COVID. Molecular tests, of course, these are the key things one has to really work on. See, I just want to change something here. So let's take a very small break before we go into the main topic. The important tests are immunological tests, which we can't ignore. This time for clotting, which I said, biochemical tests, liver function tests, CT scans, physiological tests, and then we will dive deep into our gold standard qRT. So immunological tests, IgM starts that early. Why I'm telling, no, there is a combination between this and the qRT. That's when qRT happens and when we use this. And then IgA and IgG test starts. So the antibody tests are actually the right confirmatory test for COVID-19. You can have qRT and then you have to definitely do a test. So that is the way is you can say, and also sometimes, no, you had fever, but now you're all right. But did you had COVID? You have to do antibody test. There is no other way. You can also do, it can also be used sero surveys. Now people say in Bangalore, around 20 to 25% of the people are exposed to COVID. Say you go to a shopkeeper or a guy who's a delivery boy or a cameraman, press crew, all these people will be antibody positive. We can even have a bet. We can go and check randomly 10 of them, 5 of them will be antibody positive. That's people who are exposed to others has to be positive. There is no other way of escaping. And when vaccines are coming, the vaccine efficacy, you need antibody test. So there are lab-based tests, there are rapid tests. So here is ELISA, which is done in lab, CLIA or chemiluminescent immunoassays. That's also done in lab. See here in all these cases, no, it's a signal amplification. Whereas in our qRT and see lateral flow also is kind of signal amplification. The ones which I showed, no, the same thing is here. This is IgA, IgM, IgG together, and this one is IgM and IgG separately, three different, same patient tested with four different cards. So these all work. This is a way to prove that if someone is having IgG, IgM also, then it's a recent infection. And most of these people may not even have antigen, might be minus. They may not even have COVID virus, RNA to be tested sometimes. But if they have IgM, probably they will have virus. So there are many immunological tests, rapid CD169, but it's done with flow cytometry, which you'll find any viral infection actually. Important is cell-based assays, T-cell assays, memory T-cells, interleukin assays. This is for people for prognostics, how much is the severity. Now, I hope I have told the basics so we can go into, we'll deep dive into our gold standard qRT PCR. This gold standard qRT PCR is what is widely used. Millions of tests are being done. India is doing something like 700,000 to 1 million tests per day. So, where are we? So it has 5 steps. You take a swab, collect the sample, it's a nasopharyngeal swab, collect the sample, put it in VTM, viral transport medium, which is around 2 ml, is there. Then transport to the lab. The lab force do the recording of the samples, arrange it, and then they manually transfer into deep well tubes, 200 microliters of the sample. But you see, you're only taking 1/10 of the, see, I'm just going deep so that you can understand where I'm going. You're taking only 1/10 of the sample. So you're reducing the sensitivity of your test by 1/10 to begin with. So, and then you do the extraction. After extraction, you're using only 1/10 of it for qRT PCR setup. See, this is why you know the TrueNat and others tests which are closed systems, they usually take all the sample and all the RNAs are used for it. That's why sometimes it appears as though those tests are much more sensitive than the gold standard, right? Because they are almost 50 times more of the sample goes inside a TrueNat machine, right? Or other such machines. And then you have to do a qRT PCR setup, take 10 microliters from here, mix it into qRT. So these extraction and qRT, many labs are already optimized it for the equipment. There are pipetters, low-end pipetters which can do this. And then qRT PCR is 1 hour. Then people review it. Sometimes they have to reconfirm. Maybe it's very, very close. Maybe they used one probe, maybe other probe was not good. Then you print reports. But before that, what is this qRT PCR? If in case anyone has doubts, I'm just clearing it. This is quantitative real-time PCR test. RNA becomes DNA, and the DNA is used as a template for the PCR. Typically, see, nowadays I think this one probe per tube or cybergreen based methods are gone. Okay, people are minimum using 2 probes for the virus and 1 probe for the host RNA internal control. So the qRT PCR mix contains buffers and RTP, reverse transcriptase, DNTP, stack polymerase, primers for the 3 targets, 2 for virus, right? And then the probes for the 3 amplicons, whatever PCR comes. And the fluorescent reading is taken in the each cycle from 1 to 40 cycles. So what is this is here. Here is fluorescent intensity, and the cycles is in this axis, number of cycles. And what you get, see, this is not color, but there's all different wavelengths of emission. E gene is blue, S gene is, and black is internal control. Let's say if we keep like that. So once you have the graph, this is the fluorescence versus number of cycles. Here is 40 cycles. Here is 20 cycles. So you can see here is the threshold. The threshold is nothing but above which it's noise, above that it's a good fluorescence. So you can say that E gene is around 18 cycles, S gene is 19 cycles, internal control is 20 cycles. So the cycle at which the sample exceeds the threshold is called CT, cycle threshold. So this is what will say how much is your viral load, but based on how the swab is taken, how it's transported, what volume it is, how much, how was the RNA extraction, all of them taken together, the CT might change. That's why many people say, I don't believe in CT, exact CT number. You say whether you have COVID or not. Because to believe in CT, it has to be done in an appropriate manner. But it's true that early times the CT will be in 35s, and then when you are into a higher phase, it will go down to 20 cycles, right? And then it again goes to 30 and then slowly vanishes. It all happens in a week's time. Just quickly, you might be interested to know what is inside a qPCR machine. This is a Michigan-based company which is manufacturing it here in Bangalore. So they brought their machine for testing. So I'm very quickly, so that you understand what's going on there. Say there is a Peltier block, which is important for ramping up the temperature and ramping down the temperature. So Peltier is what does that. It heats and it cools. It's one of the greatest inventions of mankind. By passing electricity in different directions, it can either heat or cool. And then there is a heated lid. It's very important. If the heated lid is not there, your sample vanishes. There is a little LED bulb on every tube, and there is a little camera which will measure the wavelength. So every cycle during extension, end of the extension, the fluorescent values are measured. So now I'm literally jumping into, see, from here, I'm going back from here, all these 5 things, I'm deep diving into each one of them to say if and how we can improve it. Okay. So I'm going. Here it is. See, firstly, VTM. What is the improvement one can do? VTM, the virus is alive, right? So folks have started using what is called MTM, molecular transport medium. Here, the virus becomes dead. It lyses the virus or it inactivates the virus. The virus is not alive anymore. And the RNA is stabilized. This is molecular transport medium. So this makes life easy. Once everything is put and maybe now it is ready to ship normally instead of taking too much precautions like in VTM. People have gone ahead and said, why this? I'll take it into trizol or lysis buffer. For example, in TrueNat case, it's taken directly into lysis buffer. So it's not like one has to go and get a approval from ICMR or someone to directly collect the sample into lysis buffer. Could be trisol. I'm just saying trisol. It could be anything. Next, instead of a lysis buffer, people can take it directly into a buffer, which is a gentler lysis buffer, and it will be compatible with qRT. I think this makes a lot of difference, because if one can avoid extraction altogether, right? You take sample, the swab, put it, swirl it into the a gentle lysis buffer, take 10 microliters and do the qPCR. Amazing, right? This is practically possible. This is an improvement which is required. See, the next is this VTM and the swab. I think one of the worst things COVID patients ever felt. I think 90% of them, the worst experience is sample taking. This is what I heard. Everything else is normal, okay? Because only some unfortunate 5% people gets hospitalized and other things come. For most of them, the swab is the worst place. Many of them didn't even have a symptom, right? So why swab? Why not take saliva into VTM? See, this is not the question I'm asking. There are methods where you can take saliva, put it into VTM and get started. It is there. People are even selling some of those kits. Why not take saliva into a lysis buffer like trisol? I don't think anyone has done it, but it's very much doable. Or why not take saliva and put it into a gentle lysis buffer, which will be compatible with the qRT mix? I think DSS Takara folks or the NeoDX people know, they have that kind of a concoction. The qRT mix itself has certain things that you can put the sample in directly. Possible. See, many times, there are a lot of things possible, but we are not doing it. So here is something I want to turn the video on the reverse side. So we will do, the swab is tedious and we can avoid it. So the next one is this transportation to the lab and extraction. I think once you have saliva or sample, if it gets into a gentle lysis buffer compatible with qRT, there is no need for an extraction, right? One can even have a 10x lysis buffer. If you have 10x lysis buffer, you will avoid even dilution of the sample. Like say you have 50 microliters of 10x lysis buffer, right? And you put 500 microliters of your saliva, 450 microliters of saliva, or even 10 microliter. So see, finally you can avoid dilution of the sample as well. Next is qRT PCR setup. Very important is automation. This is the key from beginning to end to avoid fatigue and errors. I think many of you are using it, but some of you are still doing manually, right? I know people are using manually. The problem is, even though you automate, the cost of reagents and the scale do not improve. That's the most important part. Can I reduce the cost? Can I do more? Because what is the use of automation, right? In India, we have lots of people. I don't want them to lose jobs. I can't bring automation just to get rid of people. It has to bring something substantial, right? So here is something. I'll just show you. Maybe now you can see. See this. Can you see this? See, this is the typical 96 well PCR plate, right? This takes around approximately this 20 microliter, 25 microliter volumes one has to use for doing the tests. Here is something else. I think you need good eyes to see. This is a 384 well plate. The total PCR volume itself can be only 10 microliters. Just 10 microliters one can do. This can cut cost. And this can also, doing 384 well plates can also give rise to increase in the capacity and scale, right? It can reduce cost. It can improve capacity and scale. But why people are not using it? Number one is the cost of 384 well plate. That's not a big cost because it pays for itself in one run, I think, or maybe one day's run. Like if you look at a 384 well plate machine is typically 2 times to 3 times the cost of a 96 well plate machine. Instead of 10 lakh rupees, maybe this is 30 lakh rupees, 25 lakh rupees. That's it. But many people don't have that 384 well plate machine with them. Second is, this 384 well plate requires higher end manpower and woman power. In the sense, slightly high tech. You have to use multi-channel pipette. You have to carefully do it. You cannot mix up samples because you see the plates are this plate and this plate, same thing. You have to do 4 times the number of samples, 384 and 96. But it makes a big difference. Most people are not doing because they don't have the right manpower or woman power to really do the PCRs accurately. Otherwise, one can use it. I don't know why even ICMR said, go ahead and do it. So what else we can do with the pooling technologies, right? So you can reduce the cost and increase the capacity by simply pooling 5 samples. Let's take. Simply pool 5 samples, and at 1 sample's cost, you will be doing 5 samples, right? And this will cut down your cost. It will increase your capacity, or what we call as scale. Both are possible, right? But what is the problem? Once we get these 5 samples or pools, maybe 10 of the pools out of the 100 pools are positive. Now you have to unpool them, which means you have to look at those samples and then again do it one by one. So this is going to be adding on to the time, firstly, and the effort in picking it up, and then the cost, again the cost of doing it, right? Instead of now people taking 12 hours to 24 hours, now this will become 48 hours. But still it could be good for folks for as a pooling method. But there is a better pooling, which is smart pooling of samples. That is one-step pooling. That is, you pool the samples magically after you do the tests. You will be able to say which samples are positive and what is the Q or what is the CT value, right? So this is our friends from Israel did this. I'm going to introduce you to them, that company to them. So you will directly know the infected sample, even though you have pooled. You have cut the cost, you have increased the scale, but you directly know the infected sample, right? And then here, you can pool the samples before extraction itself. So you're cutting the extraction cost also, right? But if you're using that lysis buffer, which I'm talking about, anyway, there is no extraction. See, sometimes the extraction's job itself is to compress the samples. Like if you have it in 500 microliters, if it can become 50 microliters, maybe some loss is there, it's getting compressed 10 times. But sometimes it may not be required, right? If you have a sensitive enough qRT test, you don't require it. So this is the company which we are working with called Lestes. They are in Israel. So they are looking at making the new normal a better new normal, right? Instead of waiting for a lot of time, or instead of spending a lot of monies and efforts in doing the PCR tests, can we do the same qRT sensitivity, right, and the specificity, but much lower cost, and we can process a lot of samples. See, right now, factory workers, people are traveling, they don't want to wait too long, right? So this SAMD is software as a medical device platform. They are semi-finalists in the XPRIZE rapid COVID testing. You know the XPRIZE, the same guys who are talking about $100 genome, same folks. So they reach semi-final stage. I hope they will reach finals and also get the first prize. So I will tell you what it is in just few slides. So what is this classical pooling, or called Duffman pooling? This was during Second World War times. People were that time also. See, this is not only for PCR. You can do for anything, any kind of test. Even ELISA you can pool, right? You can pool for ELISA. So here I'll show you. Here is one pool, 5 samples, and the pool is green, means it's negative for COVID. Here is another 5 samples. One of them is COVID positive. So the pool shows as positive. Now there is a step 2. That's why we call it 2-step process. So here you now go and unpool them. You go and do qRT for each one of them, and then find, hey, this fellow sample number 7 is COVID positive. This is the classical 2-step pooling. So here is now our method, 1-step pooling. So you have taken 8 samples. I'm sorry, you have taken 8 samples, pooled them, and made it into 5 samples. And this is just giving an example. Okay, but you can take 50 samples and make them into 10. Typically, or 100 samples, make them into 12, 15, because it's not necessarily you're mixing 2 samples in a well. You can also mix 3 samples or 4 samples in a well. Everything is possible. So now, in this case scenario, you can see if B and E are showing positive, then we know this sample number 4 is the one which is in these 2 wells. So this way we can actually, in 1 step itself, we can take, say, 1,000 samples, typically, take 1,000 samples, compress it into 96 samples, run the thing. So the less test folks will give you the algorithm, or to the robot which is pooling, how to pool. Actually, it is going on. I'll also show the video if we still have time. I think we have time. Then you identify which samples. This is very ideally suitable for screening purposes, right? Like you have 100 people working, 1,000 people working in a company, in a building. No one has symptoms, but who has it? This is the best way to do, right? But if in a hospital, these are COVID patients already positive, use antigen test. Don't use any test. Even qRT is a kill. You can just do an antigen test. So this can go at prevalence until 10%, 15%, maybe 20%. That much prevalence you can do. Whereas simple pooling, as per ICMR, if you exceed 5%, for example, this region, North Bangalore, is now a low prevalence of 1%. So they are allowing pooling of 5 samples. But many labs are not pooling because they are finding it difficult, the pooling itself. So what do you get out of pooling? So here is what it is. I made a small calculation. A typical extraction, I'm putting including manpower cost and all, you cost around 100 rupees. qRT mix, maybe 300 rupees per sample, and maybe another 50 for manual works. So you can operate, say, if you have 3 machines and you are running it 3 times, 4 times. You can run 1,000 samples. Whereas if you use the less tests ones, you can do 10,000 samples, and per sample cost will be 90. It's not 45. I'm just generously adding another 45 because sometimes, because mathematically you will be getting all samples unpooled in first shot. But the algorithm might return, say, please rerun these 10 samples. So I've added the cost. So at around 90 rupees, you can create you can run, you can multiply the number of throughput by 10 times, and you reduce the cost by approximately 5 times. So this is the power of pooling, and I still don't know. I still wonder why people are not using 384, because some people tried it and then said, boss, optimizing 384 is too tough, I do with 96. But if you see how much of throughput you lost, how much of monies you lost, it's really worth it. And this one is, since it's going to be, you need not optimize everything. We are going to help you with this part. We are the official partners of Less Tests in India. We are going to get you the right robotics and everything and help you with this part. So you can cut down cost, improve the throughput tremendously. And I don't want to make the next level of calculation, how much money you are going to save, right? Or how much money you are going to make, right? 10,000 samples instead of 1,000, right? What you will earn in top line in 1 year, you will get it in 1 month, right? And you still don't spend that much. So your bottom line will be tremendously high, whichever way you calculate. So it's important. I hope lots of you will do it. And you can consider these are our friends from Weizmann Institute. We are going to help them in quite a few other ways also, apart from bringing it here. So now, last slide. What and all we are doing to help people fight the SARS CoV-2. First is, we are almost 40 labs in India have done sequencing of COVID using Minion. We also supply the COVID primer pool A and primer pool B. This is called Arctic protocol. So it's important to do the sequencing, not every sample, of course, not every sample. It's important to sequence certain batches of sample, certain in quotes outbreak in certain region. You might have learned today that Netherlands is killing lots of farm animals, the minks, not farm animals, the minks, because COVID moved from that to humans with a different mutation, right? So sequencing is one part, but we are working with quite a few folks. And Tushar, I hope he's around, is pushing us to start this external quality assessment scheme. That is, this is not for manufacturers, but this is for the labs. We will send, manufacturers can also use it. We'll send the samples. You have to run the PCRs or run the assay, whichever assay it is, and send us back the results. And these are all automatically validated through manpower involved in that we do. We supply, we have already been doing it, synthetic and real standards. We supply reagents and kits because I'm connected with lots of people, including Takara guys, DSS Takara guys, NEB, Thermo folks. We have stocks and we can supply things fast. We do benchmarking of kits. I know some of you have already done it with us. Benchmarking of kits. Send your kits. We'll compare it with other kits in the market and tell you how good is yours, or maybe what improvement you may have to make, right? And what it can do, what it cannot do. We benchmark it also with different qPCR machines. We ourselves have 4 different qPCR machines. We can test in all of them. We can optimize your reagents and kits. The sequencing of COVID samples, we provide it as a service. We provide end-to-end service. For example, if you're a COVID lab, what do you want to buy? Which kit you was best? We can do the R&D part because you'll be busy doing everything. That's what I was surprised. Some 4 labs I asked. Rest of them I stopped asking because they themselves say, boss, I can't touch anything. Don't even talk about improvement. I'm just having a protocol, one kit worked, and just doing it repeatedly. So we can be your R&D partner. And finally, this is the most important part, the smart pooling by less tests. This is the real game changer. Imagine you can do 10 times without increasing a qPCR machine. All you require is one robot, and the robot will buy himself or herself in 1 month itself. So think about it. I think this will be the biggest game changer if we can bring it and start applying it. The smart pooling solution is the biggest one. Thank you, everybody, for listening, and I look forward to your feedback.